dear representatives of the national patient associations, and also general public who is interested in these issues of cirrhosis and the research that is done on liver patients. So we are very happy to welcome you to this online educational events for patients from the projects Decision, MicroPredict, and A-TANGO project. And without further ado, I would just give the floor to Professor Yalan, who will chair this meeting with me. Professor Yalan.
Okay, so look, well, thank you very much, Marco and the ELPA team for organizing this. You know, we believe that cirrhosis is really where the biggest unmet needs are, and it is really a great pleasure for me to introduce you to my colleagues who will be talking for the most part after my introductory statements. So essentially, Pierre-Emmanuel Ratou, who's a professor of hepatology at in Paris, will be telling you about the exciting project which is called Decision, which is trying to find new treatments for cirrhosis. Professor Trebecker is a world leader in terms of understanding the relationship between the gut and the liver and how it impacts. And he is running probably the largest liver study funded by the EU. All these three projects are funded by the European Commission. And he'll be talking a little bit about what the importance of the gut is in driving. Cornel Trebecker is really the world leader in terms of understanding the importance of the gut and liver disease. And he's leading the project called MicroPredict, which will define the role of the gut in liver disease. And very excited that Cornelius Engelmann, who's probably the youngest of the speakers today, is leading on a project called A-TANGO. A-TANGO, which is a combination of two drugs that he discovered while he was working here at UCL with us. And he's also won a big grant in order to bring this to man. So you'll hear three new approaches about problem solving in cirrhosis. I just want to put this out there to say that, you know, you've obviously done a lot of, you understand fatty liver disease and alcohol, but the new story today is really about cirrhosis. And this is a seriously under-resourced, unmet area. Just to give you highlight numbers, you know, 844 million people study with chronic liver disease, but of these, 2 million people die each year. And these are deaths that occur from cirrhosis. Just to put this number into context, 100 million people have cirrhosis. 10 million people have decompensated cirrhosis. And what is really frightening to us, it must be frightening to you as patients, representatives, is that mortality has remained unchanged for the last 30 years, because the only treatment we have is liver transplantation, which is available to very few. So people are dying from cirrhosis, and we need to start to think about how we reduce these deaths. And just to give you an instance, that in the last 30 years, there are only 2 new drugs that have been discovered. And these are not new drugs. They've been repurposed. The first is rifaximin, which is an antibiotic. It can't be very good for you. Ocaliva is used for primary biliary cholangitis, costs $75,000 for a year and does not improve mortality. So we are working in an area with very few treatments. And this is, you know, big problem for us. So just to give you a number that when a patient, cirrhosis occurs after 20 to 30 years of injury. It can occur in patients with any cause of liver disease. Doesn't matter whether hepatitis C or B or alcohol or fat, they all culminate into cirrhosis. And when cirrhosis occurs after 20 to 30 years of injury, 50% of these patients will decompensate, develop one liver-related complication in 5 years. And once they develop this complication, 50% will die in the following 5 years. So the clock starts with cirrhosis. And at this present time, we have no treatments to prevent these complications. We have no treatments to prevent progression. So that's a massive unmet need, which we need to try and solve together. And Decision, A-TANGO, MicroPredict are moving in the right direction, but we need more. young people to help do the science and develop new therapies. So one such group that is addressing cirrhosis, and this is a couple of words about European Foundation for the Study of Chronic Liver Failure. This is an organization that I'm now the scientific director for, and all these projects are being coordinated by European Foundation of CLIF. And as you can see here, our mission is really to improve the lives of patients with cirrhosis, not only the quantity, but also the quality. That is a key metric. How are we going to do that? We're going to do that by partnering with the best hospitals in the world. We have 170 centers distributed around the world that are starting to study cirrhosis. We are very well distributed in gender matching. We are very collaborative, and with Marco and the ELPA team, we have started to build those collaboration that brings the patients in to tell their stories. This just gives you a little sense for the kind of studies we do. We have large number of participating hospitals. We recruit a large number of patients in our trials to give them the benefit from the drugs and devices that has been developed. We collect a lot of samples so we can study them to better understand the disease. And these are the different projects that are running in CLIF at the moment. The two new ones that are not on this slide yet is a project called CHANCE, which is focused on liver transplantation, and A-TANGO, which you'll hear about today, which is focused on building this new drug for these patients. So I'm going to stop there, hand back to Marco to introduce the speakers, and we can take some questions at the end of each of these talks as we move forward. So thank you very much.
Thank you very much, Professor Yalan, for this really, really good introduction and scope of the activities that we do. From a patient side, we do understand that this is an extremely important issue. And I just want to stress one thing that is maybe not stressed enough. We are talking about ACLF. So we are talking about patients whose chances are really small, really relatively in a small range, what is there to do. So we do appreciate all the speakers that are here today with us, that are trying to change this landscape for the benefit of every patient who has faced liver disease, especially patients that are facing like ACLF or cirrhosis. Prior to the start, it's a unique opportunity to see three big projects presented in one event. This is something that was never done before, and this is something that was proposed also by Professor Yalan when we were talking, and Amelie from company Concentris. We all recognize the importance of sharing, not sharing just nice words, but also sharing the focus, the everything that we need or want change for patients. So European Commission is keen to know what these projects will bring us, what will be the benefits for the patients. And I think we have three excellent speakers or four excellent speakers that will present these projects where ELPA was invited to join as a patient representative.
Thank you for the introduction. I'm very happy to be invited to that session. I think that's a very important session. I'm really happy to present you actually what we have achieved after 5-6 years of research. And I think all the projects together, the first one and second one, our project, we all have the same aim. So we want to improve the life of patients with liver cirrhosis, and that's the very important thing. And it just tells that we're in a good way and that we are actually progressing on that path. So what I want to talk about is not only the A-TANGO project, that is the name for the overall project. And I would like to explain later on why we came to that name, why we use that acronym for that project. But I also want to talk about the substance, and that is the substance called G-TAC. And as the title says, is a really promising drug candidate for cirrhosis, and that is the novelty basically. And I try to explain you what this novelty is about and why we came to the idea to test G-TAC or repurpose the drug G-TAC for patients with acute or chronic liver failure. So you have seen similar things, and you know, I'm the last speaker, and we are all working on the same topic. So there will be some repetition, but I think it's a good thing to have repetition, because then you probably remember the main messages. So what you see here, and you've seen that in the first talk already, that is the progression of a chronic liver disease. Here's an example of the alcoholic liver disease. What happens? So if you drink too much alcohol, you have a chronic liver injury. And this chronic liver injury leads to an alcoholic liver disease, which is mainly you have more fat in the liver and you have some inflammation in the liver. And if you keep drinking, you develop a fibrosis. That means that is kind of scarring. And the more scarring you have, the more frequently you develop a liver cirrhosis. Liver cirrhosis is the end stage of that more or less scarring. It's important to mention that you have liver cirrhosis, because that might be the turning point. And the point is that all this space here, this is a mono-organ disease. So nothing happens to any other organ apart from the liver. But from that time point on, once you progress with the chronic liver disease, you will develop complications in other organs and dysfunction in other organs. So that's why it's so important to highlight that a patient has a liver cirrhosis, because that may affect other organs. You can have something like a decompensated cirrhosis. You have heard about that. So you may have complications. So you may have ascites, which is that you have water in your tummy, or you have this encephalopathy where, you know, patients behave weirdly. They can't remember things. And if you further progress, you have really an organ failure. So the brain fails, the kidney fail, any potential organ in your body may fail. And that is a term you've heard about, that the acute-on-chronic liver failure. So you have a cirrhosis, which is decompensated, and then you have further organ failures. And we are able to define at what time point or what stage you have an organ failure. And I don't want you to understand all what you see here, but as you can see, you have certain definitions for organ failure. The gray shaded areas here, and that's how we classify patients. And we can define, well, this patient has a bilirubin of 12, so we know this patient or more, this patient had a liver failure, or we measure the creatinine, which is kidney or renal parameter, so we know this patient has, if it's creatinine value above 2, so this patient has a kidney failure. And there are other markers as well for all the other organs. And what we do then, so by having these organ failures, we first of all define whether the patient has an acute or chronic liver failure. And the second step is we're combining all organ failures, and then we can determine whether that patient has different severity grades. So when you have more or less one organ failure, you have severity grade 1. If you have 2 organ failures, you have severity grade 2. And if you have 3 or more organ failures, you have ACLF grade, severity grade 3. And that's, I think that's a really important slide, because that tells you the impact of that disease. So if you have only, let's say, only this decompensated liver cirrhosis, where you have some water in your tummy or you behave slightly weirdly, what happens then? Then you have a mortality after 3 months of roughly even below 10%. But once you develop, or the patient develops, a true organ failure in any other organ, then you can see here the mortality climbs up to almost 80%. So only after 3 months. So only 20% of patients survive if they have 3 organ failures or more and decompensated liver cirrhosis. It's a really severe disease, and it tells you that we actually have to do something about that in order to reduce the mortality and to improve patients' life. So there's another slide. Again, I don't want you to understand that slide. Why I'm showing you that is I want to highlight the complexity of the disease. This is the supposed, or these are the supposed pathomechanisms which are contributing to the development and maintenance of acute and chronic liver failure. And you can see here, there's so much pathways. They're linked to each other. They're overlapping. They're increasing. They're decreasing during disease progression. So it just shows you the complexity of the disease. And it also tells you that potentially in the future, it might not be enough just to address with a therapy one of these pathways. It might be necessary that you have to intervene at different levels or different pathways, which might mean that either you have an individual therapies with different drugs, but of course you need to combine different drugs in order to avoid side effects on other pathways if you inhibit, for example, another pathway. I think that is the main message. The complexity makes it potentially necessary to use combinatorial therapies in the future. So despite this complexity, quite frequently you can highlight a few major pathways which are majorly involved in the development of the disease. And I also want, or try to show you that for the acute-on-chronic liver failure. So on the left-hand side, what you can see is what happens if you have an injury to a healthy liver. So that can happen quite frequently, and probably many of you know paracetamol. So there are many, like especially in the Anglo-American countries, there you have many intoxications with paracetamol. What happens? So if there's an overdose of that drug, you have an injury. And once you have an injury and you reduce or you take away that drug, you have repair mechanisms which are aiming for to induce regeneration and to bring the liver back to where it was before. So a complete regeneration. And you can, like in a healthy liver, you can imagine, like if you have, for example, you have an explosion in your house, it just your roof goes off. What you do, you ask the builders to come and the painters, and they just refurbish and reconstruct your house, and at the end you have exactly the same what you had before. So you have your normal liver, right? And nothing, as if nothing happened. But when you have a liver cirrhosis and you develop an acute-on-chronic liver failure, there's a difference. And just to show you that, maybe that's a bit to understand. So it's not only like one house is exploding. What happens once you're cirrhosis and you have an impact on the liver, it's a huge blast. It's like a huge fire, etc. And what happens? You can see here that remains. And you can easily imagine that even if you ask the builders to come and if you ask the painters to come, they're not able to reconstruct everything as it was before. So there will be some scarring, there will be some leftover, and probably there's even not a chance to repair anything. So these are the major aspects, in our view, of acute-on-chronic liver failure. You have an enormous injury, enormous inflammation, and on the other hand, you have an impaired regenerative capacity. So, the reason why we need to understand what's contributing to the disease is because we want to modify the disease. And everyone is talking nowadays about disease-modifying therapies as a novel approach. And actually, that's not entirely novel, but I just want to highlight why it's so important to speak about disease-modifying therapies and what it actually means. You can more or less, you can talk about two kinds of therapies. One is the symptomatic therapy, and one is a disease-modifying therapy. We are definitely, especially for that disease, we are aiming for the disease-modifying therapy. And what it means is, like, if you have a symptomatic therapy, and maybe just an easy explanation what it means, like if you have any joint pains or your knee hurts, or your grandma comes here and says, well, my knee hurts. If you do a symptomatic therapy, just ask your grandma, take a paracetamol, and that's it. But the point is, your grandma will come back the next day, the day after the next day, she will come back, because you're not doing anything with the underlying cause of the disease. So that's symptomatic therapy. And disease-modifying therapy means, actually, if your grandma comes to you, you will see, well, there's a problem. Of course, you can take paracetamol, but we need to understand why you have that knee pain, right? So you check further, and then you might discover that your grandma has an arthrosis. What does it mean? She needs a surgery because she needs a new knee. So that is actually, so you modify the underlying cause. And with that, you modify the disease, and after like 2 or 3 weeks, your grandma will not ask for paracetamol anymore. And that's what we are aiming for that disease as well. So we want to modify the disease, we want to modify the cause, we want to reduce mortality, we want to reduce the impact on that disease. And that's extremely important. And I just want to show you that graph. It's quite, maybe it's quite complex, but I try to make you understand what it means. And this is a graph from US where they try to understand the burden of the disease. And what they saw is, like, from 2001 to 2011, that there were more and more hospitalizations with cirrhosis and acute-on-chronic liver failure. You can see here ACLF is gaining importance, and there are more and more patients with ACLF. And along with that, you also have an increased, so the economical burden is increasing because the costs are increasing. You can see here, the health system has to spend more and more money for acute and chronic liver failure. So you have the mortality, you have the impact on hospitalization, you have the health burden. So this whole disease means a lot, not only for patients, for the whole community. So now talking about the GTAC, and I said we're talking about this drug substance. Actually, it's not one substance. It's actually, and that's why I was talking about like combination of therapies. There are two substances. One substance, the first one is TAC242. So TAC242 is a substance which is blocking a specific receptor pathway. So this pathway means, so if you stimulate this pathway, and that's can be done by bacterial products, etc., it actually initiates an inflammatory response. So it's contributing to the disease mechanism. So what TAC242 does, it blocks that pathway. So essentially, what it does is it stops the immune response and the inflammation. You remember the huge blast. We want to block exactly that blast. But the point is, why you have a positive effect, always when you use drugs, whatever you use, you always have side effects. And these side effects are normally negative. So they might impact even on the efficacy. And as you can see here, even the TAC242 might have a side effect. It might block regeneration. It might block the pathogen response. I mean, it might inhibit or might prevent that the organism reacts adequately to invading pathogens, for example. So there's a negative effect and there's a positive effect. So there we have a second substance, and that is the G-CSF. I'm not talking in detail about the mechanism of G-CSF, but I want to highlight what it does. So the first thing it does, it probably promotes this regeneration. So it asks the builders to come and painters to come to refurbish a house, reconstruct a house. But it also has a side effect and can induce an overwhelming inflammation. So it's not really good if you have a disease where you have an overwhelming inflammation. You have another substance which is promoting that inflammation, which is not really good. So you end up, when you have these two substances, you end up with something which is that. And the question is, what the combination of these two drugs, what do you end up with? And the idea is that you end up exactly with the positive things, because the positive effects of these drugs are actually inhibiting the negative effects of the other substance. And you end up with a synergistic therapy, which is called GTAC. We call it GTAC. So it inhibits inflammation and it promotes the regeneration and repair. So you have a novel synergistic therapy for ACLF. And these two drugs individually are not novel. We are repurposing them. We're putting them together in order to use the positive effects. And it's not that we are just thinking about these, like, it's not a hypothetical, it's not a theory that they act synergistically. We have also tested that in the lab. And that's why we are working, that's why we got to ground, because we could show that based on the lab work we did, that exactly we do have this synergistic therapy. So once you use the G-CSF only in acute and chronic liver failure, what you gain is excess inflammation. So the brown areas here, they represent like immune cells. We call them macrophages. They're invading the liver and they damage the liver because you use this G-CSF. But once you inhibit, and that's the important point, once you use this TAC242, which blocks the inflammation, this is gone. There are no macro, it's just a few macrophages in the liver anymore. And the other thing is the brown areas here, which you can see here, they're injury. So you can see this is again liver, and you can see many brown areas. So here cells are dying. But once you use the TAC242 together with the G-CSF, cells are not dying anymore. And the important thing is once you block this inflammation, you ask the liver to regenerate. So the main cells of the liver, we call them liver cells or hepatocytes, they start to proliferate. That means they divide and they repair the injured area. So you have this synergistic effect. On the one hand, you block the inflammation. On the other end, you allow the G-CSF to induce this repair capacity. And so that's the way you improve the liver in acute and chronic liver failure, and also the other organs, because they follow subsequently after the liver. What we're doing now, we test the substance in a clinical study. We use the the alcoholic liver disease, because it's a well-defined disease, and alcoholic liver disease quite frequently leads to an acute-on-chronic liver failure. And as you can see here, the GTAC, and these are the mechanisms leading to acute-on-chronic liver failure. The GTAC is meant to address exactly the main mechanisms of that disease. And you've heard in the other talks already that it's not going to be done by 1, 2, 3, 4, 5 people. That's going to be done by a whole consortium. And you can see here, so our consortium for the A-TANGO project, this is also coming from many, many countries: Ireland, Great Britain, France, Spain, Switzerland, Holland, Brussels, and Germany. And you can also see that it's not like an autocratic country where just one person is saying what's going to be done. This is like a government, it's like a parliament. You can see here many, many parties, and you can have votes on something where many committees, boards, and all can put in their meaning, and they can have a vote on many things. So this is really more like a government or a parliament, and this is supervised and coordinated by the project management concentris, and they're doing a great job in all of these projects. So this is the project itself, and normally you always have work packages in these projects, and you can see here they're dealing with different things, for example, with the regulatory aspects and ethical aspects, drug manufacturing, the trial itself. Then we collect samples, of course, like in the other projects as well, and we have to deal with data analysis and statistics. And of course, we have to think further, because in case that substance is efficacious, then we have to think about moving that forward to late-stage clinical trials and even to implement it in clinical practice. That's why we have a health economic work package. And finally, all the results have to be announced and have to be communicated. So we have work package 7, and again, project management, that is work package 8. And that's how the clinical trial looks like, and it's not very much different to other clinical trials. So I just tell you briefly how normally trials work, and exactly the same for our trial. So normally you have to check on the patient who comes in whether that patient is suitable for your trial. So you have a pre-screening period where you just assess the patient whether it fulfills the inclusion criteria. And once the patient does, you can include the patient after explaining the study and obtaining the informed consent, etc., etc. And then you have something which is called randomization, because in all these trials, it's a random thing in what arm the patient is going to be, and that means what treatment the patient will get. So we will have 3 arms, and that means 3 treatments. We have the standard therapy, that means we are treating the patients according to the international guidelines. Then we have an arm where we treat the patient according to international guidelines, but additionally, we treat the patient with type 242. And then we have a third arm where we use the combinatorial therapy on top of that guideline treatment which we have. And then we have different checkpoints where we analyze the efficacy and the safety of that trial. And the treatment is going to look like that. So this G-CSF is also repurposed. So it was used already for 20 years for other indications. So everyone knows about this G-CSF in the medical field, but so it's a subcutaneous injection, so you inject it underneath the skin. So for the first 5 days, you have an injection every day, and then an additional injection on day 8. And then the type 242 is another substance which is going to be injected as a continuous infusion for until 10 days. So this is the combinatorial therapy, GTAC, and you have a treatment period of in total 10 days. And what you have to do if you have such an early stage trial, always the first very important aspect is safety. And you have to monitor the safety continuously, actually. And always when you have an impression that there's a safety issue, that goes to the safety committee. The safety committee is assessing whether there's any safety issue. And if there's a safety issue, it has to be announced to regulatory authorities. So that is for the safety aspect, continuous monitoring. On the other hand, you have different checkpoints where you assess efficacy. And you don't do anything at these time points unless you have a major inferiority. So if the drug is potentially harmful. But otherwise, you continue the trial until you reach the end point of the end of the trial, and then you assess the efficacy, which means safety is going to be monitored very continuously, efficacy at different time points and majorly at the end of the trial. And here you can see many secondary endpoints. And I'm not going into detail. I just want to highlight there are actually three major groups of secondary endpoints. The first major group is, again, different endpoints for safety and efficacy. Another one is for the work package 6, and it's about health economy, etc. And the third major part of endpoints is actually the biomarkers, because they will, like in the other project, they will allow us to generate new hypotheses and maybe even to have ideas about potential new targets. So we're using all the patients we include and the samples we get to work further on further hypotheses. And this is the last slide. And it's, again, like what Janelle showed already, it's an impact slide, what we want to achieve actually with that trial. So before, the same before A-TANGO, what we have is we have a high mortality, many patients with partial recovery, which means they remain in a state with organ dysfunction, although actually disease is like treated already, and only minority has actually full recovery. What we want to achieve is to improve the full recovery and thereby reducing death, and to improve the recovery and reduce the burden of the disease itself. And thank you very much for your attention, and I'm happy to take questions